The easiest way to run BLAST on the NCBI is to go to the main page, select your database. If you, for example, are looking for protein, you can select the protein from the list of databases and then type the name of the protein that you're interested in. In my case, I'm going to type PCSK9 and I'm going to hit search, get the list of all databases that contain my PCSK9, which is proprotein convertase subtilisin casein, responsible for the synthesis of cholesterol. And in the list over here you see the link to the gene database, but in this case we want to look at the protein, so I'm going to select the protein from this list, which is going to show me the two proteins that are available in this database. And as you can see, one is predicted, which is probably the same as the other one. However, you see a difference in the size of the protein. So I'm going to select the RefSeq, which you see by the number NP777596.2 version 2, and you see that those are different GI numbers. So basically this is probably not curated protein. I want to select the curated protein, and once I click on it, it's going to open the description of the protein with sequences, with references when it was discovered. And over here you see the sites of protein, what amino acid it starts at, and underneath of the screen you see the sequence of amino acids that make up this protein. So in order for me to BLAST, from the top of the page you'll see on the right side analyze the sequence, and just clicking on run BLAST, it's going to bring up this number NP into the search BLAST. I'm going to leave everything without changes, but what you can see over here in the program selection, it automatically selected BLAST P, because this is a protein, and it's giving you some options to select different program for proteins. For this exercise we'll just select the BLAST P. Don't forget to click on show results in a new window so that you can have your results opened in a tab or next window depending on your settings. Usually it's going to take maybe a couple of minutes. The other way to do it is to go from the NCBI BLAST main page, and if you know what type of molecules you're looking, you can select your BLAST, either nucleotide BLAST, protein BLAST, or other options, combinations of proteins and nucleotides translated. And once you select your protein BLAST, you need to know your accession number or GI, or you need to know your sequence in FASTA format. Sequence in FASTA format is probably the most difficult. Unless you have it somewhere saved, you will not be able to type it. So in my case, I can type the protein number, which is 777596.2, using the same settings. I'm not going to change the settings. I'm going to run BLAST again. So in this window I already have the results for my BLAST, and as you can see, it's giving you the maximum score, the total score, coverage of the query, and e-value. In this case, my first hit is the gene itself, the protein itself, with the same accession number, and you see a couple of other hits which are very close, and the lower you go down, the less similarity you will see between the genes detected by the max score. You see over here the 1301 versus less than 1000 down the list. So it's showing you the main homologs for this protein, and it's showing you the similarity. As you see, many of them are predicted proteins. So in order for you to see the other species that are homologous, click on taxonomy report, and the taxonomy report is going to give you evolutionary tree that is close to your search query. Clicking on the BLAST report description will bring up a PDF file from the NCBI FTP with details on what you can do, how you can reformat it, and what kind of information you're going to get in it. So I suggest you look at this page, because it's going to give you much more information on what exactly you're looking for. It's going to give you the options that you can use in your research. This is the graphical interpretation of protein, where it's located. That's the RefSeq accession number, and that's the chromosome on which is located. So over here you can configure the genome browser the way you want to, select the tracks that you want to display from SNP subjects and some different features. Once you click configure, it's going to change over here. I enabled only the SNPs, and you can zoom in on your gene of interest, and each of the red dot or red bars are the SNPs. You see the numbers RS7606. That's one of the SNPs. That's a link for SNP summary, and that's a link for chromosome 2, and this is on chromosome 13. One of the SNPs is located on chromosome 2, and a similar SNP is located on chromosome 13. So once you click on the link to the SNPs, you're going to get to DB SNP database, and over here that's the difference why it's on chromosome 1 and chromosome 13, because one is on the sense direction, another one is a missense direction or reverse direction. You can go from here to chromosome positions, to different contigs, and you can also see the neighboring SNPs. It's a very informative database if you're looking for some associations with your disease, particularly on the gene of interest. And the analysis we're going to talk about in a different section of the class.